Hello students of grade 12, okay? Now today we're going to start a new chapter, not a new chapter, but the extension or the next topic, okay? And this will be the last video for this chapter, Biotechnology Part 1, okay? Let's try to understand the meaning of competent cells, okay? Now, since DNA is hydrophilic, DNA is hydrophilic. So as it is hydrophilic, it cannot cross the plasma membrane. Cannot cross the plasma membrane. It means that it does not allow our rDNA to enter this plasma membrane, because once the rDNA means your cloning vector plus our DNA fragments. So it has to be inserted in a host cell. Now this host should be able to accept it. Okay. Now how to provide this acceptance? Okay. The first one is said to be calcium chloride treatment. Okay, it's a divalent ion. With the help of calcium chloride, first of all, what you have to do is that you have to put in a ice, okay, ice, then 42 degrees Celsius heat shock, and then you have to put again in the ice. So this will be the method. First put in the ice, means your host cell, okay, and your rDNA. Take the host cell, rDNA, and then put in the ice, then 42 degrees Celsius, then put in the ice again. Okay, so this will be the method. With the help of calcium chloride, we have, but this one, they have to be added in a calcium chloride. Okay, the first method, in that way the cell become competent. Okay, and then they will allow the rDNA to enter in the host cells. In the case of plant cells, if you see plant cells, now if you compare the plant cells, okay, or animal cells, we call micro injection. Number 2, micro injection. So these micro injections it means that with the help of syringe, okay, we are directly introduced in the nucleus of the animal cells. Nucleus of the animal cells. With the help of injection, we are adding directly in the nucleus of the animal cells. The second is said to be, you can use the gold, okay? Coating with DNA, your rDNA coated with gold. Means recombinant if you coat with a gold, okay? And then if we allow to pass in a very high speed, okay? High velocity. Okay? We call this as gene gun method, okay? Gene gun method or ballistic method, okay? Likewise, we have, I said that we can also use retrovirus. Now, you know these retrovirus they will change the normal cell into cancerous cell. Normal cells are converted to cancerous cells. These are viruses. So if those genes that is responsible to cause virus, if we take out and then put our target DNA, then we can use this as a method. Okay, retrovirus can be used to insert into and these are usually responsible for animal cells. Okay. The next, in the case of plant, we also can use as agrobacterium tumefacience method. Agrobacterium tumefacience. Now, these are those which caused diseases in the roots, in the dicot plants, okay? Dicot plants. They cause diseases. Now what the scientists have done was, they found that this TI plasmid, there is a TI plasmid. It means that tumor-inducing plasmid in Agrobacterium tumefaciens. Tumor-inducing plasmid. And this plasmid produce tumor, and in turn, these tumors produce chemicals. And these chemicals are needed by Agrobacterium tumefaciens for their activities. Okay? So they infect the plant, they cause tumor, and that tumor produce chemical, and these are essential for the Agrobacterium tumefaciens, for this microorganism. So what we have done was, we have genetically engineered it. We have removed this TI plasmid, okay? Just say that these are bacteria, and TI plasmid was removed. TI plasmid was removed. It means that tumor-inducing gene was removed, and then we have inserted our target DNA. And this will be, this Agrobacterium tumefaciens will go and will infect the plant. So in that way we can introduce, okay, there are many processes, okay, but I have discussed four important transformation techniques, how to introduce the rDNA into the host cells, okay. Now we're going to discuss very important processes or very important techniques which we call PCR, okay. PCR techniques. PCR is said to be polymerase chain reaction, okay. It has very important significance, very important application, okay. If a person, okay, if infected with viruses, okay, or any bacterial diseases like COVID-19, okay, or HIV/AIDS, okay. So PCR is a technique by which we can able to detect or diagnose the diseases, okay. Not only the diagnosing of diseases, but they are also responsible for the gene amplification. When I say gene amplification, it means that multiplication of genes. Multiplication. If I provide you a DNA and you want to produce, and if we want to produce millions of copies, so this machine is going to help, okay, to produce millions of copies in a short period of time. So it is a polymerase chain reaction. It is also said to be thermocycler. Thermocycler. Now, to perform this PCR, it's a machine, okay? And to perform it, we need DNA template. Without DNA fragments, we cannot run the machine. We need DNA, our desired DNA, which has to be cloned, okay? Second is take polymerase. Now, this is an enzyme. There are other enzymes called as vein polymerase or PFU polymerase. So this one is not there in the textbook, okay? But these are the two polymerase which act the activity, okay? Which has the same activity like DNA polymerase. We know that DNA polymerase are helpful for the synthesizing of the complementary strand of DNA, right? So PEG polymerase, I want to give information. This is said to be these are isolated from the hot spring. Okay, these are heat stable enzymes. Heat stable enzymes. Even though you heat it for 90 plus, okay, 90 degrees Celsius, this enzyme will not lose its activity, even at higher temperature. Okay, that's why we call as heat stable enzymes. The second point is that this was the first enzyme, and because of this enzyme, the PCR method was developed. So TEC is known as Thermus aquaticus. For example, if in your DNA template ATGC, then what the TEC polymerase will do? They will provide TACG. Okay, they are going to provide the nitrogenous bases, the nucleotides. Okay. The third is primer. We know that in the natural process, primer was developed by the primase enzyme, right? But in artificial process, when we say PCR, it is in vitro. In vitro conditions. It means that in lab conditions. Okay. So this primer is designed, artificially designed. Okay, it's a short nucleotide sequence, 5 prime to 3 prime. It's a short nucleotide sequence. Okay, maybe around just say 10 to 20 nucleotides. 10 to 20 nucleotides. While designing primer, two things are very important. It should be short, point number one. Okay. The second is it should contain GC. Okay, GC content should be less in the case of primer. Now, we also need DMTPs and some magnesium ions. Okay. The first step for PCR is, this question came even in your CBSE examination, the last year examination, this year examination. Okay, PCR question was asked for 5 marks. Okay, so the first step is said to be denaturation. CBSE paper 2020. So in denaturation, if this is your 5 prime and this is your 3 prime. Now remember that is an automatic process. In a machine, it has already set up denaturation. Okay, denaturation is a process, the first step, where the target DNA will be splitted into single strand. For example, if there is a adenine, then there is a thymine, guanine, cytosine, thymine, adenine. So there is a hydrogen bond, right? ATGC, triple bond, then double bond. So it will get separated, 5 prime to 3 prime, 3 prime to 5 prime. Just said that here. It means that if the double stranded becomes single stranded, it means the bond is broken. Okay. Why is it broken? Because in denaturation step, it is a heating process. And then here we have set up the machine for optimum is 92. Okay. Optimum temperature. And normally for some microorganism, 94 to 96 degrees Celsius. Okay, but the optimum temperature is 92 degrees Celsius. If you kept the DNA at 92 degrees Celsius, then double stranded will get separated and they will form single bond. So that is what happened in the process of denaturation. Second is said to be annealing. Annealing. The meaning of annealing means, see, once the DNA is separated, has formed single stranded, right? Now, what will happen? Primer will come, okay? So just say that primer is 5 prime to 3 prime, right? So the primer will come and bind. So here is a primer, so 5 prime to 3 prime. So this is our primer. Primer will attach. Primer will attach. So annealing usually happen at just say 50 to 60 degrees Celsius. If it is at higher temperature, then primer will denature. So high temperature is not required, and not more than 65. Okay, some books will say that it is 50 to 60. Some will say 66. Okay, but normally it is 65. Okay, and for some microorganism it is 50 to 60, but it should not cross 50 to 65. Okay, because primer will denature. For the process of replication, if the primer has to come first, right? Because without a primer, DNA polymerase cannot come to its activity. Because its activity is 5' to 3' direction. Now once the primer comes, we call next step as extension, the third step, extension. Extension means, for the extension we need what? Tag polymerase. We need DNTPs. We need some magnesium ion. So this tag polymerase will provide, now they will provide the strength, okay? Extension of this strength. Now this is just one cycle, okay? One cycle. Denaturation, annealing, extension. Automatically in a machine, this process will keep on going on. So if you want to produce, just say 2 into 10 or 2 into 4. It means that 2 into 2 into 2 into 2, right? 4, 8 into 16. So you can produce 16 copies within a minute, because denaturation is happen in just 1 minute. Okay? No, and then even for extension, it is only for a few seconds. So in just a minute, you can produce 2 copies. Just said in 1 minute, you produce 2 copies of DNA fragment. So in a short period of time, you can just multiply the cycle. For example, you want to produce billions of copies, then you can produce, just say 10 cycles, 20 cycles, depending on our desired DNA fragments. Okay? So this here is a in vitro condition. In a lab, we can also produce DNA fragments in multiple copies. Once these DNA fragments are ready and introduced into the host, then the next challenging process is bioprocessing. Bioprocessing or downstream processing. Once the protein that this DNA fragment will produce these proteins, just say insulin protein, then we need to produce in a large scale, okay, with the help of industries. And for that, we need a vessels for production, just say millions and billions of insulin, useful for the diabetics patient. And you want to produce cancer medicines, okay, then we have to produce in a large scale, and this can be done with the help of bioreactors. So bioreactors is a vessel which is going to produce the desired products which is beneficial for the human being, okay, in the form of medicine, in the form of vaccine, okay. So bioreactors are the vessels that will produce your products in a large scale. And once it is produced, it has to be in a market. And for that, you have to purification. First is separation. After separation, you have to go for purification. Then packaging. Before packaging, put some, if it is a food product, put some additives. The last one is commercialization. And if it is a drug, commercialization. If it is a drug, we have to go for testing. So, with this topic, I want to complete the chapter, the explanation for the biotechnology. In the next video, in the next class, we are going to discuss the part 2, the application of the biotechnology. Thank you.